All right, good morning, everyone. How can everybody hear? Can everyone hear me okay? I can hear myself great, so I don't know if I need the rest of you. So I'm going to be talking to you today about creating and sustaining diverse study enrollment. So we're going to be talking about what I'm calling engagement and recruitment science. Hopefully, the ideas here will not necessarily be novel to most of you. These will be things that you have done in each of your study sites, each of your institutions, each of your companies. But hopefully today what will be new is stringing it all together in a way that makes intuitive sense for a workflow-based approach. So we've got four main goals today. We're going to be talking about why diversity is important in a research context. We won't be spending a lot of time there because hopefully by this time in 2019 we recognize its value. We'll be talking a little bit about the history of recruitment science. I'm going to be giving you just a little tiny hint of some preliminary data from my research center on what recruitment science might look like. And then finally, for those of you who are just very, very concrete kinds of folks, we will have some very detailed real-world advice for how to bolster your recruitment, sustain that recruitment, and ensure that it is inclusive and diverse in its approach. In terms of major disclosures, there are no relevant research disclosures, but my research center does engage in a large amount of community service in Boston, which is partially supported by Otsuka and Merck. So let's start with the importance of diversity in research. I start with a quote that I think when it comes to those of us who talk about social justice in the healthcare realm or even healthcare adjacent realms like clinical research, this is almost a cliche, but it's really important to keep in mind. Of all forms of inequality, injustice in healthcare is the most shocking and inhuman. So these words were uttered by Dr. Martin Luther King Jr. During the Selma marches back in the 1960s. But what many people don't know is that he was referring quite directly to the Tuskegee syphilis study. This was well known by the black communities throughout the 50s and 60s, and yet there was nothing they could do about it until the New York Times published a single article in 1972 that resulted in the study being shut down within a few weeks. Previously, though, the study was run for about 40 years, from 1932 to 1972. Many of you in this room have at least heard of the Tuskegee syphilis study. But what I want you to keep in mind over the course of my talk today is that there were a number of innovations and advancements in the way that we conducted the Tuskegee syphilis study that are still in play today. There were a number of innovations such as return of results, such as study incentives, and the way that research participants were encouraged to be retained in the study that we still do today. They have their roots in the Tuskegee syphilis study, and so we need to keep that in mind as we're trying to break free of those particular sets of biases. Before I move on, however, I just wanted to draw your attention to this intentionally poorly designed slide so that you are aware that the Tuskegee syphilis study was not the only significant medical abuse against the black community in the United States. The era of scientific racism was invented here in the 19th century in the American South, where it spread north, and there were a number of research studies that have been conducted up until quite recently. If you can see that bottom one, that date is well after a lot of the human protections research protections were put in place. So in Columbia University in the 1990s, there were further abuses, suggesting that we can't just rely on IRBs, we can't just rely on the Belmont report to save us. We need to be very thoughtful in our approaches without thinking necessarily about compliance regulations. So the Belmont report was a direct response to the Tuskegee syphilis study. The final report was published in 1979 after the commission had been meeting for about 5 or 6 years. And we are all deeply familiar with the three main pillars of the Belmont report: respect for persons, beneficence, and justice. I would say that in the intervening years, the few decades since the Belmont report, we've gotten really, really good at understanding what respect for persons looks like, what beneficence looks like, but it has been a little bit one-sided, to the extent that we are still struggling to ensure that our research studies are truly just. So let's talk a little bit about why diversity is crucial for clinical research. Now, there is a very strong and significant social justice argument to be made. I'm not going to be making that argument today because I hope it is well known to this entire audience. There are other reasons why diversity is crucial for clinical research, and they pertain directly to our bottom line. So number one, up to 20% of FDA approvals, so this is after we've gone through the primary clinical research stages, phases 1 through 3, there were differences, significant differences, in either the exposure or response of devices and therapeutics as a function of racial or ethnic group. 20%, 1 in 5. So if you just do basic statistics, if you've worked on, let's say, 10, 15, 20 protocols in your career, there are really strong odds that a drug that has made it all the way through the first 3 phases and has presented for FDA approval may still have significant differences in how it performs in the wild. Now, those of you might say, well, that's what phase 4 monitoring is for. That's what post-approval monitoring is for. However, we also recognize that at the very earliest stages of our research, of our clinical research, there are really significant representation issues. And that in our most cutting-edge research studies, there are particularly acute problems. So when we're thinking about precision and personalized medicine, when we're thinking about leading cancer studies, there are really foundational representation issues at the genomic level. And my next slide will kind of cover that in a little bit more detail. But I did want to highlight some really great work that was published in the past few years by Elizabeth Rose Maeda and Jennifer Weave, who talk a lot about an epidemiological approach. Now, those of us who are clinical trialists, sometimes we feel that there's a tension with the epi folks, but what I'm here to say is that we need to understand that our work needs to scale to the same extent as we see with the epidemiological approach. So even though we are conducting primary research, we need to ensure that the populations that the epi folks that are studying for a particular disease presentation are the ones that we are serving as we design our clinical research trials. So that's really crucial to keep in mind because, as I said, the way that we conduct research studies with a focus on efficiency really pushes us to recruit samples of convenience. So we recruited samples of convenience back in the 1940s and 50s and 60s, and those were largely disenfranchised, poor, or black individuals. Sometimes Hispanic and Latinos were included as well. Obviously in World War II, Japanese Americans were included in that as well. Now, since the passage of the Belmont Report, the definition of a population of convenience has changed. Those groups are now much more privileged than previous to the Belmont Report. However, we still have selection and survival and recruitment biases to our research studies. And if we are not modeling that variance, for those of you who are stats folks in the room, if we're not modeling that variance, what are we doing? We're throwing it into our error terms, which means that it's harder to detect any significant effects because we're throwing a lot of information that we could know away. Now, this is related to that second bullet point. So Latrice Landry and colleagues last year published a really phenomenal article in Health Affairs that I encourage everyone to go out and read. So if you look at the GWAS catalog, as well as the other relevant catalogs for a number of clinical research studies, if you look at the genomic presentation, you can see that it's not terribly diverse. And I want everyone to keep in mind that as we're marching towards more personalized and precision therapies, these are the foundations that we're relying on. The vast majority of research is conducted on Western European whites. We have seen an uptick in recent years as work out of China and particularly India has come on board, driving some diversity in those genomic databases. But keep in mind that that third dot, that orange dot, is everybody else combined. You can see in the GWAS catalog, it's not great. We can see that we're doing some work in respiratory, but we don't have any Asian representation there. Ultimately, we have an enormous diversity problem in the way that we're conducting foundational and basic research that we cannot overcome at the post-approval phase. How do federal research guidelines encourage diversity? There's a lot of stuff going on. There's actually been some recent draft guidance from the FDA as recently as a couple of months ago. However, what I want to encourage you to think about is think beyond what the regulatory guidelines are. Those are in place. That is certainly a minimum standard that we should, of course, ensure that we are compliant with. However, let's talk about diversity in clinical research. So this is the latest FDA clinical trial snapshot. This is just table 1 from their summary report. And what you see is that we've got some diversity. And if you're looking at those numbers and you're comparing them to what I've presented below in white, you might think we're doing pretty okay. We've got a lot of women in research. We've got better representation of racial and ethnic minorities. But again, I was trained with like a very heavy statistical focus. We know that averages ain't everything. So we know that average is a measure of central tendency. I would also encourage you, when you're thinking about these things, to look at elements of dispersion, which is to say, let's look at variability between studies. One way of understanding whether the mean is a good representation of the information that you're gathering is to look at another measure of central tendency called the median. Okay? So if we look at the median, we see that the numbers in the clinical trials snapshot are quite different. The median representation of women in research studies is not that 56% that we thought we were seeing with the clinical trials snapshot. It's closer to 48%. We see that this is particularly egregious when we're looking at racial and ethnic minorities, where the median representation of Black and African Americans is not that 11% that's implied with this table, it's actually 4%. Asians, Hispanics, and even older adults are not as well represented as we would think. And for those of you who are really, really nerdy, and I hope there's a couple of you in the audience, I did also calculate the coefficient of variation because, why not? So the coefficient of variation is a measure of variability. I did write a dissertation on this, so I'm happy to walk anybody through it later if you want. The coefficient of variation is a great estimate of variability within a number of observations, and it tells you how wonky something is, like how much it kind of jumps from one number to another. And you can see that the numbers for women and whites are relatively stable. The numbers for Black and African Americans, Asians, Hispanics are much more variable. As you approach one and exceed it, you're talking about studies that are not at all consistent with one another. So even as globally we march on towards better representation in our research studies, we see that on an individual study basis, that's kind of that's the level at which many of us operate, we're not doing a great job. So let's talk a little bit about early efforts in recruitment science. Most of this is dedicated to me sort of being a fanboy over a particular paper that came out earlier this year by Milo Rasuli and colleagues in 2019. They published this in Genetics and Medicine. Again, fantastic paper. There's a lot of really great information that you can dig into. But ultimately, they said, we've got 4 years of data. We did 7 different recruitment methods. Which one was best? Which one is the best source of recruitment for a genetic study? So they did 7 different methods. They relied on their team's staff network, so word of mouth. They did community-based events. They did 2 different kinds of flyers, one that was targeted to a certain populations, one that was very generic. They recruited directly from a clinic. They did a biobank data poll, and they also pulled from electronic health records. What we see here are 4 different colors. The blue bar refers to the people that they reached out to and were able to screen, and then it kind of goes through the recruitment workflow. The red bar is basically who was able to kind of come in and give them that baseline data. What you should see here is the power and the seduction of EHR-based and biobank-based data polls. So we can reach thousands of people with that EHR, but if you look at that red bar, what percentage of those folks are actually getting in the door? Relatively small. Now, as you might imagine, a clinic-based sample does have a relatively high rate of return, but this is a really great thing that we can easily implement to understand the efficacy of our recruitment efforts. Now, Milo Rasuli and colleagues don't stop there. They actually also mapped the proportion of those who were enrolled to those who were invited. And what you can see here is something that's not as evident on the previous slide, where you can see that community-based events, which are often regarded as something that we do as long as we have the time or the money, something that's regarded as optional or ancillary, in fact, yields a very high proportion of those that we touch. On the other hand, the biobank-based and EHR-based recruitment efforts are much more low yield relative to those that we actually invite to participate. So this is not based on that initial screening number. This is based farther downstream in that workflow to those who we actually were able to contact and invite to participate in a study. So again, think about the way that you conduct recruitment and retention within your own clinic practices, within your own clinical studies, to really determine what's going on with your research participant population. You might find, like Milo and Suli colleagues did, that the events that we often dismiss in terms of their efficacy, or the ones that we struggle to sustain, are truly effective. Number of folks getting in the door is not the only interesting measure of recruitment. We also have to think intensely about cost. Now, this particular research team did something that was interesting. So in addition to calculating the actual dollars that they had to spend to get somebody into a research study, they actually incorporated staff time. So number of phone calls, time spent writing emails, times chasing people, around a parking lot to get them in the door. And what you find is, again, surprisingly, that biobank-based and the EHR-based data recruitment methods were astonishingly expensive per participant compared to some of the other methods, especially staff network, clinic-based, or community-based recruitment. So again, we should be very careful not to be seduced by that huge blue bar that I showed a couple of slides ago that said if we do an EHR data poll, we can reach 3,000 people easy. What we fail to incorporate is that in order to get even one of those individuals in the door, it will cost our staff hours of their protected time, and it will be egregiously expensive compared to other methods. So if we're thinking about recruitment and retention, especially with the eye on diversity, we often think of one thing and one thing only: does it scale? If you present these data looking at the cost per participant, I think it's a little bit tougher to argue that any kind of recruitment method would scale with the costs that we see with the traditional recruitment measures that we have here. Finally, and most relevantly to my talk, we have to discuss diversity. Which methods yield a diverse sample? Again, this is an intentionally poorly designed slide. What I want you to keep in mind is that we have a big red rectangle with pluses and minuses. If you see a plus, then a particular recruitment method is good for that group. If you see a minus, we don't really know. The only thing that you need to take away from this slide is that it's inconsistent. Diversity is not a monolith. If you want Latinas in your study, you don't do the same thing that you do to reach individuals who are in rural populations. If you want Black and African Americans in your study, you don't do the same things that you do to reach out to individuals without a high school education. You need a diversity of methods to bring in a diversity of research participants. So now I want to turn to some preliminary data, very, very preliminary data, very early data from my research center, the CARE Research Center based out of Boston, to focus on diverse recruitment. And these slides are not designed to be comprehensive. They're really designed to kind of show what if, just to kind of be a little bit tantalizing. And I'm happy to talk a little bit more about some of these data after my formal presentation if people are interested. So when we talk about early efforts in diverse recruitment, we need to understand operationally that there is a difference between diversity and inclusion. Diversity refers to the principle of trying to make sure that as many different voices are represented and heard as possible. Inclusion, on the other hand, really refers to a design that says that any voice that wants to speak up has an opportunity to do so. So when we think about diverse recruitment, we think about diversity in clinical research, a lot of early work goes back to Badal, which was, obviously a therapeutic that was designed for Black and African Americans. Actually, for those of you who are kind of a little bit more nuanced in their presentation, this was designed for African Americans more than it was for Blacks. And if you don't know the difference of those, come and ask me and I'll explain it to you. So what I want to encourage you to think about is not mere diversity in your efforts, because at the scale that we have to operate, especially in this day and age, it is not possible to reach all audiences, especially if you think about an intersectional approach. So not just Black people, but Black men in particular. That's a different approach. Better to think about inclusion. So the state of recruitment science is, in a word, fragmented. There have been numerous peer-reviewed manuscripts across every discipline. I'm sure some of you in this audience are well published in the state of recruitment science. However, there are no clear definitions, no consistent definitions. There is no data framework or any kind of ontology. And there's a really, really strong lack of comparative effectiveness research. The Milo Rasuli paper that I showed you just a few moments ago is about the most comprehensive comparative effectiveness study that has ever been published on recruitment science. Most articles that are published focus on single case studies that are usually nested within a single protocol, and they are focused and report only on enrollment. There's very few metrics of recruitment, very few metrics of outreach, and even fewer metrics on engagement. There are huge gaps in the literature that have not even been acknowledged, let alone addressed. And then the conclusions tend to towards advice that is really difficult to operationalize, replicate, and measure. How many of you have thought about a recruitment measure and then you've talked to somebody about it and they said, oh, we've tried that? How many of you are subject to the we've tried that effect? So somebody says, I got this really great idea. You go out and you give everybody gift cards to CVS and they will come in droves. And you find out, well, actually, we should be giving out gift cards to Walgreens. There is no way within the literature right now to operationalize that difference. And so we end up subject to the we've tried that effect, where we've tried something, but we don't understand the nuances well enough to understand why something was successful or why it failed. So what that means is that there are lots of confounds within recruitment science. So there is also this effect that's sort of on the opposite of the we've tried that effect, whereas there's the charismatic, underpaid individual effect, where there's somebody who's just really, really good at reaching a particular population. They are not necessarily paid to do that. They don't necessarily have protected time. And when they move on, usually because they are clinical research coordinators, they'll move on to medical school or graduate school. Recruitment tanks, and we don't understand why. The second point, I think, goes without saying. What is a minority even? A lot of the work that I've presented so far has focused on racial and ethnic minorities, but that's not the only folks that are underrepresented in the room. If you aggregate clinical trials, especially the history of clinical trials, we recognize that they are really well designed for people who are white, male, high income, overeducated, usually very well educated, and they live in urban centers along the coasts of the United States. If you are not all 5 of those things at once, you are a minority. So I don't know if anybody's willing to recognize that they're a white, rich dude in the room, but if you're not that, then you're not well represented in research. So you are all effectively minorities. And we need to really understand recruitment and retention science, as well as the engagement processes in order to make sure that that works. We often fail to deconfound available from accessible, and there's a really great example of that happened today. So those of you who are in the front of the room have a very different auditory experience than those of you who are in the back of the room. I don't know if there's anybody who switched between Dr. Lau's talk and mine, but there is a huge difference. But from my perspective, all of you are equally available to hear my words of wisdom. However, the truth is that my words are not equally accessible among you. So for those of you who are having hearing difficulties, I encourage you to move to the back of the room where the auditory experience is a little bit better. There is a huge conflation of outreach, recruitment, retention, engagement activities. People will do an engagement activity and expect changes in recruitment. People will do an outreach effort and expect changes in retention. We cannot continue to confound these things. Infrastructure is largely absent. It's usually down to a very dedicated team who don't have enough protected time. They're usually not paid very well. The study staff is rarely dedicated purely to recruitment. And finally, that last point is really, really salient. Engagement and recruitment activities are usually entirely separated from most study protocols. They are not well integrated in terms of a workflow. Now, why can't we recruit diversely? Why can't we recruit inclusively? These are the 10 biggest reasons that often come up. We don't need to go through all 10, but I'm sure that some of you have come up against 1 or maybe even 10 of these in your regular research recruitment activities. We're going to come back to these 10 in just a moment. What my research center does is we believe that there needs to be a workflow-based approach to engagement, recruitment, and retention in research. Fundamentally, what we often do is we focus on the screening, randomization, and retention aspects of our research studies, and we ignore all of the first 4 steps. And when we end up coming up with barriers to enrollment, barriers to screening, we find that it's because we have neglected some of these earlier stages. I'm going to explain a little bit about what I mean here in just a few minutes. So if you think about diversity as a workflow problem, or as I like to think about it, workflow as a diversity problem, what you can see is that bottlenecks using this workflow-based approach can occur at multiple stages. So you need to start by defining your sampling frame, which too few of us do. We will define our catchment area, and we'll say, we are living in this very dense urban center that's full of people, but we don't fail to capture, we do fail to capture who within that dense urban center is actually able to access our research studies. Then we usually have an awareness problem. Almost all clinical research studies are not penetrating into the general population well enough, which is why we tend to have huge recruitment problems. There is no engagement or trust determined. This is usually manifested as a lack of return of value. This is why people tend to be apathetic towards research studies, because they don't understand what science is, much less clinical science that involves poking them. So there are a number of implications to presenting diversity within your workflow. And so there should be a very clear differentiation of the kinds of activities that are conducted along this workflow. So outreach is primarily at the very earliest levels. Recruitment is somewhere in the middle. Retention is a little bit towards the end of this workflow. And then finally, the appropriate characterization of participant availability is the primary barrier. I don't know if any of you have heard of something called Lasagna's Law, but it basically says that we wildly overestimate how many people are willing to do our research study. And then that number sort of availability dwindles as we get through our workflow and start to panic. And then as soon as we're done with that protocol, we go right back up again with the number of people that we think can join our studies. This has been well defined. It is not merely restricted to thinking about diversity and inclusion. This is just how we tend to do our science. And this has been well characterized for about 50 years now. So let's rethink those 10 barriers that I presented earlier that are really well characterized within the research literature. It turns out that you can map these things really well onto a workflow-based approach. So lack of awareness of research opportunities, which is a main driver, maps really onto awareness and the engagement factors, as well as mistrust, which often is thrown around as a major reason why particular subgroups don't tend to participate. There are some barriers that tend to map onto multiple stages of this workflow. And then similarly, there are stages of the workflow that map onto multiple barriers. So we can recognize that the screening barrier is a really significant one for a number of reasons, which is why single solutions to the screening problem don't tend to yield large results. And then finally, the rest of these map on relatively nicely to the other workflow areas. So what you see here is that as you address earlier barriers, you might be revealing ones that are downstream. And then if you try to attack a downstream barrier without being aware of earlier ones, then you may not see the kinds of results that you're expecting to see. This is what drives the "we tried that" effect. So leveraging the framework within work that we've done at the CARE Research Center, we recently completed a multi-year, multi-site observational study of cognitive impairment. We did it at about 59 sites. We were kind of embedded within a large neuroimaging protocol where there was relatively low minority recruitment, in this case referring to racial and ethnic groups other than Western European white. We solicited proposals about 18 months ago for a 4-month intervention. Ended up being a 6-month intervention in the end. We selected 4 of these 59 sites from all over the United States. We gave them roughly $40,000 to try to address the problem. And we made them do monthly dashboard reports and phone conferences to keep us in touch of what was going on. What's really key to remember here is that we allowed the sites to identify the principal barrier that they were trying to address, as well as design an intervention with our close collaboration. And so what we have here is actually fairly simple. So most of the sites identified barriers outside of their typical research workflow. So they had challenges in the awareness of the study, challenges in trust, challenges in interest, and they worked on dedicated interventions that were designed to address those. The interventions were focused usually on engaging communities. Again, communities is defined quite broadly here. So one site engaged with minority-serving physicians as their definition of community. So we have to keep in mind that it's not always about reaching patients and prospective participants directly, but sometimes those who serve as gatekeepers, and in this case, minority-serving physicians. We monitored sites from roughly August to December of 2018. We also checked back in with them over the last 6 weeks. 3 of the 4 sites had actually returned back to their baseline pre-intervention engagement activities, so they weren't able to sustain this effort beyond the money that we were able to give them. And then the last site actually had used that money, leveraged those funds to develop and sustain a really great relationship with a Spanish-speaking clinic for Hispanic and Latino referrals. And this is it. This is kind of the nuts and bolts. So in the 12 months of 2017 and 2018, all sites, again, all 59 sites in the United States recruited about 50 underrepresented minorities. In 2018 to 2019, that was about 35. So they actually got worse during the intervention period. The pilot sites, the sites that I just discussed, between 2017 and 2018, really radically improved the recruitment of underrepresented minorities. In this case, we are referring to Black and African Americans, as well as Hispanic and Latino individuals. And so if you want to do the really quick math in your head, that yellow bar on the right is effectively about a third, a third of the bar that's on the right of the blue site. So between 2017 and 2018, what we ended up doing was our 4 sites were driving about a third of all diverse enrollment for this neuroimaging protocol, again, with a very simple 4-month intervention. We also managed to boost study-wide recruitment from 10% to 12% in that same period of time. But what we noticed is that the most successful pilot sites shifted the recruitment burden from the underrepresented minorities and assumed it as a study team. So trust was less of an issue than we expected, and the establishment of an engagement metric separate from enrollment was really crucial, because as we all know, what gets measured gets maintained, what gets measured gets cared about. And what we found is that when there were deliverables that were separate from pure enrollment or even screening metrics, it was a lot easier to sustain and justify activities. Still, with this particular design, there were a lot of challenges that remain. The most notable challenge is, can we actually do this in a way that really creates comparative effectiveness? Can we scale this? And then, again, what exactly is a minority? Is there any room for thinking about minority much more broadly? Because, again, thinking about Blacks and African Americans, thinking about Hispanics and Latinos is going to be wildly different than thinking about individuals of low SES, individuals who are living in rural areas, et cetera. So can we systematically address these barriers much more broadly in a comparative design? And so that leads us to the second study that I want to talk to you about that we are currently conducting in conjunction with the Michael J. Fox Foundation. So Michael J. Fox Foundation launched a platform called Fox Insight some years ago. It was designed to address all of the barriers that we've discussed so far. It's a completely online study of Parkinson's disease that theoretically should be available to anyone with an internet connection. So here, you should finally see a huge diversity of enrollment to Fox Insight, because anybody, especially those living with Parkinson's disease or Parkinsonism, can sign up. They don't have to go to a brick and mortar site. They don't have to go to New York to the Michael J. Fox Foundation directly. They can sign up from their phone or their tablet or their computer. However, what we see here, and please do not adjust your eyes, is that when it comes to diverse enrollment for this particular study, it's not terribly diverse. For those of you who can't read the labels, the graph is white for a reason, because what we see here is that enrollment is about 90% white. And it's not just racial and ethnic diversity that's a problem for this digital online-only study, is that it's, if you look at annual income, keeping in mind that median income in the United States is around $50,000 to $53,000, most individuals in this study, I would say like a plurality, earned over $75,000 annually. And you can see that the biggest bump was for those earning $100,000 a year or more. So we're looking at an online study that is designed to have no barriers whatsoever, attracting very wealthy white people. Again, if you think about other aspects of diversity, we see that when it comes to education, there is a bump for the bachelor's degree, but there's also a bump for master's degrees. And there are so many overeducated individuals with this study that there are different categories for professional degree versus PhD or equivalent. So what we're seeing here is that even though, again, the study is designed to be very inclusive by being online only, it's still only attracting the same old folks. So we designed a study that's really focused, again, on those aspects of the sampling frame: awareness, engagement, and interest. And what we found is that we decided to focus on multiple barriers. What we ended up doing was we're trying to look at cost. We defined minority very, very broadly, well beyond thinking about race and ethnicity. And then we had a strong focus on defining a very clear, detailed protocol, as detailed as any aspect of our clinical workflow, but just focused on engagement and recruitment. That's all it took. And so what we ended up doing is we have 8 sites in the study. 4 of them have been randomized to control. We gave about $45,000 to the intervention sites, $10,000 to control. All sites developed their own workflow and intervention around this. And what we found is that the interventions tended to focus on community populations as well as physicians. The pre-intervention launched formally at the beginning of this month. I don't have any actual data yet from this particular study, but what I wanted to show you here was this is how comparative effectiveness research in a science of engagement and recruitment ought to be designed. So we do allow some flexibility because we recognize that sites are different, which is a little bit different than how we often think about clinical research. But we do focus on comparative effectiveness. We do focus on detailed protocols. So what's next for science of recruitment? As I mentioned, validating some of these metrics for each workflow stage. There are metrics that are already broadly available, thinking about epidemiology, digital platforms, market research. We heard about the need for professionalization and integration of expertise in these phases along all these stages. As I've been hammering home over the course of this talk, we need comparative effectiveness research, and we need to continue to examine this workflow in lots and lots of other environments. There are some really revolutionary ideas in there, but for reasons of time, I'm going to not get into those today. So I just wanted to provide just a few detailed pieces of advice for people. If you want some broad advice, if you want to try to solve this problem directly without being dedicated to the science of it, you can just think about the step 1 is separating that catchment area from your sampling frame. Very few of us do that, and it always is going to present a problem. Be thoughtful about quick fixes. You probably have an awareness problem that needs fixing. If you want to get new people in your study, you have to do new things. And then keep in mind that the CRCs, the RAs, the ones that we often term worker bees, tend to have an expertise that we don't, the people who are more seasoned in this field. We need to trust them. And if you are going to be seeing some of the talks along track 5, there are a couple of great talks about social media and digital engagement that I think are well worth your time. If you need concrete advice, and I'm going to try to wrap it up here quickly, use plain language for everything. I couldn't have had a better introductory talk to kind of really highlight how incredibly important this part of it is. Language equity shouldn't be the problem it is. It is a problem, but even within English speakers, let's try to get it right. People really aim for a 5th grade reading level. There are lots of readability calculators available online for free, and it's not just about vocabulary, it's about embedded clauses, it's about sentence structure, it's about terms of art and acronyms. Use clinician champions and research ambassadors. Please don't tell me you've tried that, because if it doesn't work, you haven't done it well. Design for a strong return of value. This was implicit in Dr. Lau's talk. If we cannot convince people that this is worth their time, then they should not be getting involved in research in the first place. And then sustained community entrenchment works, but it takes time. The magic number that I've seen in my few years of doing this seems to be around 7 years. So there's a little bit more concrete advice here. I'm just going to leave this up, but I'm going to try to wrap up my talk now. If you've got your phones out, maybe take a quick picture of this slide. This is not stuff that should be rocket science or unknown to any of you, but what's really, really incredible is to keep in mind that we need to be measuring these efforts, and we need to match our outcomes to our interventions. It's the same rules that apply when we're doing any kind of clinical research. We need to be applying to a science of engagement and recruitment. All right, with that in mind, I'm going to go ahead and wind up there. So thank you so much for your time.